Okay, so now I'm recording. So the review session is just to work through any questions that you have. I'm not going to stand up here and lecture. So I think you got my announcement email of what's going to be on it, right? So we have the light reactions. This is the slide set that it starts with. And then, no, it's just this slide set and part of the next one, right? Because that covers before and CAM and all that stuff beyond it. So that leaves us with the light stuff, light reactions, T3 cycle, PCO cycle. That's it. So, question on the light reaction up there. It's like this slide set, so I think it starts posted as light reaction for photosynthesis. So that PowerPoint like that. And so it'll be all of this material. So that excuse me. We're working through all of that. So it's a big slide set. All right. And then the next slide set is the carbon fixation. And that one will work through the material through the PCO cycle. So then the material starting on my slide set is slide 40 with the C4 cycle. That stuff forward we won't be doing on this exam. So I sent out an email. If you check your, excuse me, if you check your email. Oh, I haven't talked much today. So what questions do we have on this slide set? The Q cycle. Okay, so I'll start here. So with the Q cycle, we're looking at the point right in here as we're looking at the interaction of the plastoquinone that was associated with photosystem 2. Once it grabs its 2 electrons and 2 protons, it's going to release and move laterally through the membrane until it can bump into cytochrome B6F. Then, once that happens, it's going to donate its 2 electrons, but they go in 2 different pathways. One is going to go linearly, which is that second bullet point. So we'll hold off on that. And then the other one goes into the Q cycle. And so to kick that off, it's going to get, the electron's going to get past the cytochrome B. And here it is visually. So it gets past the cytochrome B, then to another cytochrome B. And then there's a plastoquinone there as well. But as we know, plastoquinone isn't chemically happy until it can get 2 electrons. So now all of that has to repeat all the way back to the very start of light striking the photosystem 2, the chlorophyll electron getting excited and getting another PQH2 to come through again. And then when that happens, so here's the second one now. The second PQH2 that releases off of photosystem 2 finds its way to that same cytochrome B6F and it donates its electron into the Q cycle. So now you see that semi-reduced plastoquinone now gets that second electron, grabs 2 protons, and releases as the fully reduced PQH2 And in that process, a couple of key things, right? So first we have the two PQH2s that release off of photosystem 2 and migrate over to cytochrome B6F. They're dumping their protons. So in both, so here is put together the first PQH2 on top, the second PQH2 on the bottom. So you can see there's protons getting dumped, so to speak, into the lumen by the first PQH2. And then the second PQH2 comes along, dumps its protons, and then that plastoquinone that's semi-reduced gets the second electron, grabs 2 protons. So when you add it all up together, you see protons disappearing from the stroma in the sense that they're getting bound up into the PQH2, and then you get protons being dumped into the lumen through the action of this Q cycle. So that's describing a proton gradient across the thylakoid membrane. That's a good thing. What does that lead to? Mm-hmm. No, so yeah, so here is what we consistently see as the PQH2s are doing their thing is that you're going to get protons disappearing from the stroma. So even back here with the PQH2, when it's sitting there waiting for its electrons from photosystem 2, eventually it will be grabbing protons from the stroma. And then the Q cycle, you see the same thing happening there. Protons are getting grabbed from the stroma and getting put into, reacted with PQ to fully reduce it to PQH2. So in those cases, it's disappearing from the stroma and then getting dumped into the lumen. So you get this buildup of protons in the lumen and less in the stroma. So that will get you your proton gradient. And so they're trying to show that here as well, where you see protons getting pumped across from stroma to lumen. Water is releasing protons in the lumen. So you get this buildup of protons. Mm. Yeah, I guess as it's disappearing from the stroma, dumped into lumen, you might tack in your own phrases to clarify that. Yeah. Yes. Yeah. Yes. So when you think about a proton gradient across a membrane, whether it be the thylakoid membrane or the mitochondrial membrane, whatever, and that should get you immediately going, oh, proton gradient, that's potential energy. So now in this case, the chloroplast, the plant can do something with that. And very often what the strategy is to get ATP made. So those protons will diffuse back through the ATP synthase. And as they're flowing through that, it generates the torque force to allow for ADP and inorganic phosphate to react. And that gets you ATP. So here they're trying to show the protons flowing through and then the synthesis of ATP. So that's one of the huge payoffs of doing the light reactions, is to get that ATP. Mm-hmm. So with the details given on the slides that I've typed out are, so let's go to, typically we always start talking about the light reactions from photosystem 2, just like how we did in class. So that won't change. So if I start asking, assume I'm talking about light energy coming into photosystem 2, what happens? And so that's what's being shown here on this slide, that P680 asterisk means that when you write it like that, it means that the chlorophyll electron got excited in the reaction center. P680 refers to photosystem 2 reaction center. And then the first acceptor in the reaction in photosystem 2 is pheophytin. And remember, you can, if it's an essay question, you can abbreviate all of this. You don't have to type it all out. And so that electron getting transferred from that chlorophyll in the reaction center to pheophytin, that is where you actually see, that's where they can point and say, oh, that's where light energy got converted to chemical energy, because you actually had a passing of an electron from one molecule to the next. So that's how, you think about it kicking off. And then we talked about the fact that you need, to replace that lost electron. That chlorophyll's missing an electron, that's not good. You need that electron replaced. And so water is utilized for that purpose. So the oxygen evolving complex. So I would like, OEC, you don't have to write it all out, but you could say the OEC is the site in photosystem 2 where you have water oxidized, getting its electrons stripped, somehow phrasing it so that you understand electrons are coming off of water to get past to that chlorophyll that's missing an electron. So the level of detail that's being shown in these slides that I've typed out, that's what I'm looking for in terms of the process. So for example, if you keep going on, that pheophytin is going to give an electron to the first PQ that's sitting there. And then that one quickly gives it to the second PQ. And then I say repeat. And what that means is you go all the way back to the very beginning and start this whole process again, so that, because you got to have that second PQ getting one more electron in order for it to be stable, and then it'll grab 2 protons from the stroma. So that level of detail, understanding the process of electron transfer from donor to acceptor on down the line. And then the idea of, where everything is located. So you have some big complexes, photosystem 2, photosystem 1, cytochrome B6F and ATP synthase. Those are massive. They span the whole width of the membrane. They don't really move. So it makes sense that you have to have some mobile carriers. So understanding the difference between the big complexes, the mobile carriers, where are the mobile carriers? Are they in the stroma, lumen? Are they in the membrane? What other questions on the light reactions? Yeah, so we talked about that idea, how in, we show you that beautiful little mock-up drawing, but in reality, when you look at the actual chloroplasts, in reality, they do kind of sequester where certain complexes are located. So we talked a little bit about that, how you have the light harvesting complex 2, you can see kind of light purple is more in the stacked region of the membrane. That's the granum region. And then the photosystem 1 tends to be more of it is in the stroma lamellae. So it's, but of course, when we're, you know, in order to talk about it and have it make sense, we tend to have everything neatly lined up, but in reality, it's not quite that neat. So this slide, let me find it. Where is it? So this is slide 38 on my slide set. I think this gives all the players. Goes field fight, and they do PQA, PQB. I say first PQ, second PQ. And then that second PQ, once it gets 2 electrons, they don't really show that it's fully reduced, so it's not quite as accurate, I guess. But and then you have the cytochrome B, cytochrome B. They kind of confuse up the plastoquinones here a little bit, but it does have all the players pretty much. And then they have all the players listed out here in the photosystem 1, all the way to iron sulfur B. So this kind of gives all the players. I think it's a little sloppy on some of the details. So that's why my text slides are important, that you're following those for the detail. Yes. Basically just knowing that another way to talk about the integral membrane proteins is transmembrane proteins. I probably use that word more than integral. And so when we say integral or transmembrane proteins, we're just talking about those 4 big complexes, and that's it. Mm-hmm. No, so I like this slide because it's showing they're better than the one I was just showing you, where I said it's a little sloppy on the details. I like this one because it's showing you that A0, A1, all of these are part of photosystem 1. So that, the first, so when that P700 chlorophyll in the reaction center gets excited, you can see it's giving it off to A0. So that's the first acceptor. And then it gives it to A1, then iron sulfur X, iron sulfur A, iron sulfur B. I just like the visual of it, that you can tell that those are all within photosystem 1. And it's not until you get to ferredoxin that you get out of photosystem 1, so to speak, with ferredoxin being a mobile carrier. So I think it has some nice visuals. And then it also shows a nice visual of plastocyanin coming along and donating an electron in to replace the one that got lost by light excitation. Are you talking about the Calvin cycle, or where are you? Oh, the damage going on? Yeah, so trying to get into some of the research they're doing with what happens if there's too much. Oh, okay. Are you into the other slide set? Okay. Okay. Ah. What about some of the terms? What's linear photophosphorylation? So linear, what does that mean? Straight, right? We're going from point A to point B, so linear. So that's referring to the electron flow. Because we talked about how sometimes some plants might do this little circular flow. So please try to get this straight in your head. Linear means straight line, point A to point B. Has nothing to do with circular. Okay, I've seen students confuse that in previous semesters, and it's just stress. It's just exam stress causing you to kind of invert those two. So linear means we're having electron flow from, ultimately all the way to water, from water, all the way through until we end up at NADP+. So that's what they're referring to with the word linear. And then photophosphorylation, photo, light energy, is being used. And then phosphorylation, what gets phosphorylated that's a big deal during the light reactions, during the process? What gets phosphorylated? ADP, right? So ADP gets phosphorylated and you get ATP. So this phrase is just trying to get you to think about how is ATP getting made? And they gave it this fancy phrase, okay? What they're trying to say is that as the electrons are moving all the way from water to NADP+, as that's all happening, it creates a situation that allows for ADP to get phosphorylated, get you your ATP, and it's all done because of light energy kicking that all off. Okay, so make sure that you're clear on this phrase. I've seen time and again, I'm not quite sure why. I think it's just stress during the exam and you tend to flip and I'll see students start talking about circular. There's no circular here. There's nothing to do. Linear is in the phrase. Okay, linear. So when they think about linear photophosphorylation, it's just this idea that as you have electrons flowing, I didn't get into all the thermodynamics of it, but at key steps in that electron flow, there's a little bit of free energy that can be used to pump protons or get protons, getting dumped into the lumen. And that's building that lovely proton gradient. So it's allowing for the protons to build up, and then you get that flow of the protons through the ATP synthase. Other questions on the light reactions or anything else in this slide set? So the pigments toward the start of the slide set, we got into I guess probably the most detail I'll ask is that that there is C and D also, so we've been talking a lot about chlorophyll and A and B because they're involved in the light reactions of higher plants, and that's what we tend to focus on in a class like this, but being aware that C and D are also options depending on the whatever photosynthetic organism. But knowing precisely that diatoms, dinoflagellates, and brown algae have chlorophyll C, I'm not going to get to that level. So, but knowing, yes, there are different versions beyond chlorophyll A and B, and it depends on the microbe or the plant or the algae. No, I won't ask dates. Red and blue really is what I'm looking for, that you understand, and you don't have to memorize exactly what wavelengths those are. This diagram here is, I guess, one of the concepts on this that would be good to make note of is that when the chlorophyll pigments absorb blue light, it actually excites them more than if they absorb red light. And so knowing that basically that you have these different wavelengths of light and they can impact the chlorophyll a little differently, but red and blue in general are the two favorite wavelengths or quality of light that plants will maximally absorb. And that's been shown in figure B here. So whenever you see that lovely absorption peak like that, that means that they're absorbing those qualities, those colors of light, which means they're using that red and blue in this case for photosynthesis, which is to excite the electrons, to get water stripped of electrons. And then the other qualities like green, you can see is in between the two red and blue area, and green doesn't really get absorbed. That's just bounced back to our eyeballs. That's why leaves look green to us. They don't really care about green. They want red and blue. I won't be asking about fluorescence and phosphorescence that's being shown on here. The triplet state would be a good thing to know, that that's the most stable state of electron excitation, and it does allow, hopefully, for the transfer of that electron to the acceptor molecule. Should we shift over to the carbon fixation? Okay, let's go there. We can always come back to this, but I want to make sure we have enough time to cover the carbon fixation. So carbon fixation, now we're thinking about part 2, phase 2, the biochemical reactions, sometimes they're called. And so looking at carbon fixation. So here they're showing both linked together, where you have the light reactions on the left and then the carbon reactions on the right. Remember, I don't like the way the artist did this so much, because I don't want you to think that the Kelvin cycle can take place during the night. Remember, it can't. So and so I'll refer to it as the C3 cycle or the Kelvin cycle. That's usually how I phrase it on exams. But carbon fixation, whenever you hear that phrase, what they're saying is you're taking CO2 and you're fixing it into a sugar, some kind of carbohydrate. So that CH2O, so that chemical equation that I have up here is how I'd like you to think about it, rather than the other version that we tend to learn in middle school, the C6H12O6. That I prefer not to have you think about that one. Think about the idea that we're building CH2O, carbohydrate, and glucose is definitely not the first product. And so that older equation makes you think about glucose. And I don't want you thinking about glucose. So, Calvin cycle. I will be talking about this pie chart. I'll call it the pie chart if I refer to it. And, here you can see there's 3 steps to it. So if I say what's happening during the carboxylation step, you got to be able to orient your brain to, okay, what part of the pie chart is she talking about? Okay, so same with reduction, regeneration. So you need to learn it in context of looking at the pie chart, and this is a nice kind of big picture overview of what's happening, especially when it comes to the regeneration phase. There's a whole lot going on there that's not being shown. The carboxylation and reduction actually has quite a bit of detail here, at least to the level I want you to know it. They're just missing some of the key enzymes. So think about the pie chart and the 3 different processes of the pie chart being shown there. So it's broken down that way in the typed up slides as well. Yes, did I see a hand somewhere? No. Peripheral. Any questions on the carboxylation step? What's the acceptor molecule of when CO2 comes into the plant? What reacts with CO2? What's the molecule? You can just say the abbreviation. RUBP. What's the enzyme that facilitates that reaction? Yes, Rubisco. So please, please, please do not get RUBP and Rubisco mixed up. You got to get that locked into your head. The way I always remembered that is the CO. The C stands for carboxylase, O for oxygenase, ASE, that's the enzyme. RUBP, on the other hand, is a 5-carbon sugar. So looking at that first reaction, the only enzyme I want you to know is Rubisco. You don't need to know the other ones. And then we get into, once that's done, we get into the next phase. So we just talked about carboxylation to get you that PGA, get 2 identical molecules of PGA. So that's the carboxylation reduction. Any questions on that? So again, with this one, knowing PGA feeds into a reaction where you're going to end up with G3P, but you have now also the ATP and NADPH feeding into that reaction. So now we finally see where those molecules come in handy from the light reactions. So that's showing PGA on the left, and then on the far right, you got G3P. Again, lock that into your head. I've seen students confuse those 2. Nope, I don't care about the middle one, other than it gave people a lot of headaches back in the day. They didn't know it existed. So it caused a lot of confusion and really slowed down progress. It's very, very unstable, very quick to break down. So they missed it with their technology at the time. So that's where Melvin Kelvin, he got the Nobel Prize for that one. What about regeneration? What are we regenerating? Yeah, RUBP. So, any questions on that? Basically, the regeneration, you're starting with that G3P pool. That's how you should think about it. It's starting with that G3P pool. And then you see a whole lot of stuff happening. Now, that would be, I'm not really going to cover that till the next exam. The whole conversion to DHAP, and then it goes out for sucrose synthesis. I'll talk about that on the next exam, the 4th exam. So with the regeneration, just focus in on how do we combine these G3Ps in playing that numbers game in order to get finally ribulose 5-phosphate. Yes. No, I'll get that on the 4th exam. So we'll think about that on exam 4. So the export idea or the conversion to starch, that we won't see that on exam 3. And so remember, you don't need to know all the names or enzymes. It's just the idea of the numbers game. So this slide set is posted as well, where we go into the numbers game. So you do have to tell me what 3-carbon means. What do you mean by that? What molecule are you talking about? So you have to define that. So I know that you know, but then you can just play the game of combining these G3P molecules. And the magic number is 5. That's what we're trying to create here. And so you don't have to memorize all the words or enzymes. It's more of this last step. I do need you to know that all those 5 carbons do rearrange toward the end there to give you ribulose 5. So that's last in purple and red and green there, that very last part, that you need to know. And again, that ATP is needed. What else? Yeah. So the very last step. Where'd it go? Oh, so there, yeah. So you see ATP coming in right here to take ribulose 5-phosphate and make it RUBP. Need one more phosphate to stick on to that molecule so you can get RUBP. What else in this slide set do you have questions on? Or do any of you find that one useful? I put it in there just for studying purposes in case your brain works with it, but Yeah, I know. It's like you have to pick it apart and dissect it. Yeah, I much prefer that one. Just a little much easier. And then that one too. This one's a little bit better than the other one, but it's not a whole lot. Does this one help with anybody? You do like this one? At least you can carbon count, do it that way. So, but it is pretty busy. And they color code it. So they show, blue is carboxylation and green is the reduction. And then I guess the tan or pinkish tan is all the regeneration. What other questions might you have on this slide set? Yes. Oh, PCO. PCO. All right. So what's the enzyme we're thinking about? Rubisco, right? Yep, it's kind of a relaxed enzyme. It says, yeah, I can carboxylate, but sure, I can oxygenate. And so again, we're thinking about what's going on around Rubisco. Is it seeing lots of CO2? Then the Calvin cycle is going to cook along just fine. If it, for some reason, has low CO2 and enough O2, then oxygen is going to win, come into the active site of Rubisco. And that's a pain because, well, you get one PGA, so that's good. And that can feed right into the C3 or Calvin cycle, but that phosphoglycolate's only 2 carbons. So knowing this first reaction for sure, and then understanding what happens to the 2 products, the PGA, yeah, I can go right into C3 or Calvin, but that phosphoglycolate, that's where you start kicking into the PCO cycle. And so you got to convert that 2 carbon phosphoglycolate, go through all that mess of the PCO cycle twice essentially to get another PGA to form. So it's a lot more effort to get one more PGA out of this process. So again, with the PCO cycle, knowing how it kicks off. So we just went through that, and that's actually being shown on the top left here in this figure of in the chloroplast. You have the REBP and oxygen giving you a PGA, which they swoop over to the right here. And I drew in the C3 cycle right there. So that PGA goes right in to the C3 cycle. And then the phosphoglycolate, you can see you have to follow out of the chloroplast into the peroxisome, mitochondria. And actually that has to happen twice so that you can get enough carbon. So knowing what's leaving each organelle. So knowing that by the time the phosphoglycolate leaves, it's in the form of glycolate, then it has to, then what's the next organelle? So peroxisome. And then you go through reactions to create glycine. Then that moves to the mitochondria. And then essentially that all has to happen one more time because you need 2 glycines, which they show here in the mitochondria. Here's one of the glycines on the left, and then they're showing off on the right the other glycine. So that those 2 glycines are coming from that left part of the pathway running twice to get 2 glycines down in the mitochondria. And that's just so that you can have enough carbon to create this little reaction here to give you serine. The serine's 3 carbons, okay? And then that's not the only thing of importance. Mitochondria, you also see that CO2 getting lost or respired. It's not going to get used by the chloroplast for the Calvin cycle. And so it's considered a loss of carbon. So knowing that that happens, knowing where it happens in the mitochondria, okay? So then serine leaves the mitochondria, finds the peroxisome, gets converted to glycerate. So now we're close. So now if we can get that glycerate back to the chloroplast, expend some energy, use some ATP, then you finally get one more PGA molecule that can get fed into the C3 cycle. So this black and white picture is what I tend to teach off of, but this is also an option. And I guess this can be helpful because it's showing carbon counting, plus they nicely say that you have to have 2 glycine in the mitochondria to get this reaction to convert 2 glycine to serine. I wish they would have put that CO2 like big noted box around it or circle around it or something, or bolded it, because that's a big deal. That loss of CO2 is a big deal. On the what now? No. So like, let me show you. So like you see these reactions going on here in the peroxisome. Yeah, don't worry about those. Don't worry about the intermediates. You don't have to worry about, NADH is getting used here. ATP getting used in back up here is important. That's important. But other than that, and then the loss of CO2 in the mitochondria. So those 2 things are really, but knowing what's going on here in the peroxisome with the swooping circle here between the 2 reaction pathways, no, you don't need to know that. So definitely an extra energy cost. It's a lot, pretty complicated. 3 different organelles, extra ATP needed. It's a lot of effort. And to summarize it, this slide is summarizing it, what's going on with all of that. So if you run it twice, that means you'll get essentially 4 carbons converted into 1 more PGA, but then we lose carbon in the form of CO2. But 75%, I guess it's better than nothing. Certainly better than 50%. So, but in general, when they do experiments on these plants, if you force the plant to run the PCO cycle a lot, they don't do well. They suffer. Their biomass crashes. They don't get nearly as much biomass by the end of their life cycle. And so they generally consider it, not such a great thing for a plant to be running continuously or frequently. So that weird enzyme, Rubisco, just never seemed to evolve a high binding affinity just for CO2. It seemed to, well, back when it was first evolved, there was no oxygen to worry about. So that they think was a big reason why it doesn't seem to tell the difference between CO2 and O2. But you'd think after a couple billion years, it would have figured it out through natural selection. So that makes some scientists say there's a reason for it. We just haven't figured out exactly what that is. And so they're working on that. They published a couple of recent papers saying Rubisco's not so bad after all. They actually have some nice hypotheses. So, but in general, at least experimentally, if you force a plant to do PCO a lot, they don't do well. What else? We got, so 26, looking at this one. Yes. So for the enzymes in the Calvin cycle need light. And the reason they say that, it's being shown here, for the enzymes work off this principle where basically you need to get ferredoxin in its reduced state. Well, how do you get ferredoxin into its reduced state? You have to run the light reactions in order for ferredoxin to get reduced. So going back to this, here you see the light reaction process and you see ferredoxin as a player accepting an electron. So during the light reactions, its job is to give an electron to the FNR mobile carrier enzyme, but sometimes it's needed to run this so that those enzymes in the Calvin cycle can get up and running. And so this is just the ferredoxin is trying, kicking off this really short redox reaction. So ferredoxin donates the electron to this ferredoxin thioredoxin reductase, and that will break those sulfur bonds when that accepts the electron. And then that enzyme will give the electron to thioredoxin, break those disulfur bonds, and then the thioredoxin will interact with one of those 4 enzymes of the Calvin cycle and get it activated. And so it's just a short little redox, boom, 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 in order to get one of, those 4 enzymes activated. So that's why they say they're light activated, because you have to have reduced ferredoxin kicking it off to start. What other enzyme is light activated? Rubisco. Yeah, don't forget about that. That's a different process. So what's being shown? How is it getting light? Why is light necessary? Can you tell by the drawing what's going on here? Mm-hmm. Yeah. Right. So you got that proton gradient developing through the light reactions. And that, of course, will shift your pH in the lumen. So as all of that's happening, it creates a situation where magnesium can then have the ability to flow out and find Rubisco and attach to that activator CO2. So Rubisco actually interacts with 2 CO2s. It'll take one into its active site and react that with RUBP, but then it also needs a separate CO2 to do this. So this is the separate CO2 that you see on this diagram. Okay, so this CO2 right here during the carbamylation step, that is the activator CO2. That's not the one docking into the active site so it can react with RUBP. They're not even showing that one on this picture. I haven't finished making it. But it'll be similar. I'll be able to tell you more probably Tuesday or Wednesday. I'll have a better draft of it. And it'll be separate again, like multiple choice, true, false, and then the essay, short answer. Friday at noon. And then I can stay a little bit late because no one's in there after us. Photosystem what? Oh. So we got the light reactions. Damage. So damage, we got so to deal with the damage, too much light can be bad, coming in too fast. So really easy way to try to deal with that and try to minimize impact is to create a situation where it gets released as heat rather than developing a situation for free radicals. And so that can be done with carotenoids. So carotenoids actually run a short reaction, and you don't need to know this little reaction here of the violozanthin, anthraxanthin, zeaxanthin. I'm not going to ask about that in any detail, but if you understand the idea that this is a short reaction going on with the carotenoids that allows them to dissipate heat rather than creating a free radical. So the carotenoids, pretty much any photosynthetic organism that they've ever studied, they all have carotenoids. It seems like it's a kind of a universal mechanism, pretty straightforward mechanism to try to get that intense light coming in too fast, get it dissipated as heat. And then we got into, they're focusing in on photosystem 2 and photosystem 1. I'm not really going to ask you to define or like memorize photoinhibition per se, just understanding that too much light energy causes damage, all right? You don't need to know the word photoinhibition, but too much light energy causes damage. They've even narrowed it down that the D1 protein seems to be the weak link. That one gets hit first and damaged first. And you know, it can get reversed, the damage can get fixed as it's happening, but sometimes it's too much and then they have to totally take out the D1 protein, synthesize a new one. And then photosystem 1, again, just knowing free radicals, that you can get free radical formation. You don't have to know superoxide and all of that. Free radicals, and that they're not a good thing, damaging, and that, you know, we have certain enzymes too that help with free radical damage. So do plants. So again, ferredoxin here is playing a role that's not so good. So far, we've been saying how cool it is. Ferredoxin is in the light reactions. Ferredoxin activates some of those enzymes in the Calvin cycle, but here, not so good. Ferredoxin, whoops, gives an electron to oxygen. Not such a good thing, because then you get free radical formation. And then understanding just the idea that you want photosystem 2 and photosystem 1 to work in concert with each other, because it's a linked linear flow of electrons. You need those two working together or in concert, but that doesn't always happen just because of the way in reality they're all laid out in the thylakoid. Then you have to think about sunlight striking the leaves. Some leaves are going to be shaded, some are in bright sun. You got a whole messy situation going on there, so it's not always exact. And so they have to have a way to kind of deliver energy to the one that's lagging down, boost them up with a little bit of extra energy. So again, it's just it's a phosphate group that's really telling the plant what to do. You have any questions on that information being shown here? It's the light harvesting complex. That's what LHC stands for. Those are the antenna complexes that kind of surround photosystem 2. And then sometimes they're right around photosystem 2, but in this case, if the LHC2 antenna complex protein is phosphorylated, that actually tells it to disassociate from photosystem 2 and find photosystem 1. Basically, it's trying to get photosystem 1 to kick into high gear and get up to speed with what photosystem 2 is doing. So the plant is assessing PQH2 levels. That's really how it's doing it. It's saying, okay, if I have a lot of PQH2, that's back here. So we're thinking about photosystem 2 is working just fine. If you end up with a lot of PQH2, that means something downstream of it is lagging. And that's probably because photosystem 1 isn't keeping up to speed with what's going on back here with photosystem 2. And so basically, if PQH2 levels are building up, that pretty much means that photosystem 1 isn't getting hit with it's not getting hit with enough light energy. Does that make sense? So it's kind of like backed up. It's like, I got nowhere to go. I can't deliver my electrons through to plastocyanin because plastocyanin is bogged down waiting for photosystem 1 to get excited. It's like, come on. And so then, so the PQH2 levels start to build up, build up, build up. And that's the signal. Oh, okay, then we need to get more light energy over the photosystem 1. What else? Any other questions? Today's Sunday. Exam's Friday, so you have time. So if you're sitting there going, ooh, okay, I'm really having to dredge this from my memory, hopefully this is the stimulus to get you going, get this firmed up. You have, several days now to get this firmed up. Hopefully this helped you discover your strengths and weaknesses and got you thinking. All right. I can give you more information on, the details of the exam. Like I said, probably Tuesday or Wednesday, but it's going to be a similar format in general. Okay. All right. We'll see you tomorrow.